So now we're going to talk about how to detect and quantify gene products. A lot of stuff we've talked about so far has been focused on DNA, okay? How to edit it, how to quantify it, how to sequence it. So now we're going to switch gears a little bit. So I want to bring your attention back to the central dogma. As you can see, this should start to feel like a running theme. And we've been largely focused here on the DNA. But what about the products that come from DNA? And so what I'm talking about here is we can look at levels of RNA. So after DNA is transcribed into RNA, how much does a cell make? Or we can look at the proteins that follow. How much of a protein actually goes from RNA to the polypeptide? Or we can even, you'll start to see with Westerns, we can look at sometimes if these proteins are modified with phosphorylation and things like that. So let's look at ways to detect these products. So these are blotting methods. And it was first discovered by Edward Southern. Okay, so Edwin Southern. So he called it the Southern blotting. Okay, so it was named after him. And Southern blotting is used to detect DNA. But with all the new technology, it's not used as much anymore. Then, after that, sticking with the Southern part, they come up with Western blotting used to detect proteins. Northern blotting used to detect RNA. And there's even an Eastern blotting that you don't hear too much about. So we're going to talk a little more about Western and Northern blotting. And Western blotting is largely still used today. Okay? And the techniques you'll see between the two, they follow very similar steps. So with the Northern blot to identify RNA, first you have to take this living sample, okay? Extract that RNA and purify it. Then you're going to run it on a gel. And so we've talked about gel electrophoresis, how it's going to run according to size and from positive to negative. Okay, so this is separating our RNA molecules by size. Then you transfer it. So you're going to take the samples that have run on the gel and transfer it to a membrane that you can then use to label and detect. Okay, so the gels, when you touch them in lab, you see they're like flimsy and things, and you can't do what you need to do when the RNA or even the protein is stuck in this gel. So you transfer it to a membrane that's easier to work with and lasts a little longer. After that, you're going to probe it. So remember, this is all RNA. These are sequences. So you have a DNA probe that if it's complementary, then it binds to that RNA. If it's not, it's washed away. Okay, and so then you can then take that and see what it looks like. You can see where those bands are. So here is a representation of it. This is Northern blotting. This is looking at tropomycin. And we see that different cells have different size and quantity of tropomycin. And so if you look at the size of the band, you see that there's more in muscle cells than in brain cells. But you also see that they are different sizes. Okay, so the further down they are, and then the higher up, this is showing you that there is a change in size. Now, what you will see is that smaller ones tend to go further. And what this also shows you is that because they are different sizes, they're alternatively spliced. Okay, the actual fragment of that RNA is different sizes. So there's a change in size that is usually accomplished by alternative splicing. So that's the Northern blot used to detect the RNA. The Western blot is used to detect protein. A little more complicated process, okay? So like if you look here, this is everything. And I know at first sight, this picture is like, whoa, what is going on? So let's try to break it down. So similar to Northern, you have to extract the proteins. But here, you don't have to purify them. You have to denature them, okay? So you're going to treat it with a detergent to denature it and give it negative charges to allow it to flow. So then we go ahead and we're going to separate it in our gel. But here again, our gel is made of polyacrylamide. And then after you run it on the gel, you transfer it to a membrane. So this is either PVDF or nitrocellulose. After you get it on the membrane, then you block it. Okay, so you're going to coat that membrane with BSA, so bovine serum albumin or milk, And what that is going to do is help you to get rid of non-specific binding. And then you treat it with an antibody. So this primary antibody is going to be specific to whatever protein you're looking at. Okay, so you let it sit in this primary antibody for about an hour at minimum, and you're letting those two things interact. Once it's attached, it will stay there, and then you'll wash it to get rid of any that is not attached. Okay, so after several washes, then you treat it with a secondary antibody. Now, this secondary antibody is usually labeled, so it has HRP, and then it's going to bind to the primary antibody. So usually, if this primary antibody was made in rabbit, okay, then your secondary antibody is going to be able to detect rabbit. So secondary antibodies are not as specific, so it's not a one size like the primary, but any that are in rabbit can be recognized by the secondary. And then you wash it again, and then you incubate it with some chemolucents, which is going to let that HRP show, okay? And so way back when, you used to do this in a dark room, and you would put a film down, and then that bright band that illuminates is going to be picked up by the film. Now there are other ways to do it, and so that you can use colors or different things, and the machine picks them up for you. But either way, you have to detect those bands. But what you get is very similar to what you've seen with the northern, okay? And so this is taken from a Western blot, and here you can see the levels of tubulin that show up in different cells. Now here you see they are all about the same size, but the darker the band, the more that is expressed there. And then another thing you have to do with Westerns is have a control. And so this is a loading control, and this is allowing you to know if you have the same amount of protein in each gel. Okay, so your bands, the color won't mean anything if you have overly saturated one with protein and then the other one has less protein, because now what you see is not actually how much is there, but it's something from human error. So this serves as a loading control. Okay, so this is GAPDH, is ubiquitous, all cells have it of roughly about the same amount. So this serves as your loading control to say that you do indeed have the same amount of protein, and you do quantify the protein to make sure that you are loading the same amount of protein in each one. Okay, and then you have a ladder that helps you to figure out if this is the correct size. Okay, and so this is the Western, again showing you everything. You extract the protein, denature it, run it on the gel, blot it. You can also stain it with Ponsu S, which will let you know if the Western worked. Okay, so it's just going to go and bind to all proteins there, so you can see, yes, this worked. Wash that off, and then do your primary, secondary, and look at it. But that's the Western blot. So what you should see is that they are all roughly the same. You have to extract whatever it is that you are working with, and then you either purify or denature it. You put it in gel electrophoresis, you transfer it to a membrane, and then you blot however it is that you blot so that you can detect it. Okay? And so if you have any questions or anything on this, let me know.